Plate counts are commonly used to determine the number of bacteria in food and milk. Plate counts are based on the assumption that microbial cells present in a sample mixed with melted agar medium will grow into visible, separated colonies, each derived from a single cell. An advantage of the direct plate count is that it only measures the number of viable cells in a sample. That is, only the bacteria that can grow on the nutrient agar are counted. We're going to count the number of viable bacteria on alfalfa sprouts. 1 gram of food may have 10,000 bacteria. This could produce up to 10,000 colonies on a plate, clearly too many to count. Therefore, a sample is diluted to get a countable number of cells. Serial dilutions occur when the same dilution factor is used for all of the dilution tubes. The dilution blanks contain 9 milliliters or 99 milliliters of sterile distilled water, or 0.85% sodium chloride. 1 milliliter of sample transferred to 9 milliliter gives 1 milliliter of sample in a total volume of 10 milliliter. 1 over 10 can also be written as 0.1, which is equal to 10 to the negative 1. 1 milliliter in 99 milliliters is a 1 to 100 or 10 to the negative 2 dilution. If we transfer 1 milliliter of the 10 to the negative 1 dilution to the 99 milliliter dilution blank, the total dilution is the product of the two dilutions. Remember, you add the exponents. Label the 9 milliliter dilution tube 10 to the negative 1 and label the 99 milliliter bottle 10 to the negative 3. Aseptically weigh 1 gram of sprouts. Try to get the measurement as close to 1 gram as possible. For solid samples, microbiologists equate 1 gram to 1 milliliter of a liquid. Remove the cap from the tube and use your forceps to transfer the sprouts to the 9 milliliter dilution blank. Recap the tube after the sprouts have been transferred. Mix the contents using a vortex mixer. Hold the tube at an angle. You're trying to knock bacteria off the leaves, not dissolve or suspend the leaves. Or alternately, you can create a vortex by quickly flicking the tube with your index finger. Using a sterile pipette, aseptically transfer 1 milliliter from the tube to the 10 to the negative 3 bottle. Unwrap the pipette and attach the aspirator. Use the pipette to measure 1 milliliter from the tube. Uncap the 9 milliliter bottle and transfer the contents of the pipette, and then recap the bottle. Mix the contents by shaking the bottle 20 times through a 35 centimeter arc. Label the outer, bottom edge of 4 empty sterile Petri dishes. 0.1 milliliter of 10 to the negative 1, 1.0 milliliter of 10 to the negative 1, 0.1 milliliter of 10 to the negative 3, 1.0 milliliter of 10 to the negative 3. With a sterile pipette, transfer 0.1 milliliter of the 10 to the negative 3 dilution to the appropriate plate. Using the same pipette, transfer 1 milliliter of the 10 to the negative 3 to the appropriate plate. Using the same pipette, transfer 0.1 milliliter of the 10 to the negative 1 to its plate. The same pipette can be used when going from the lower dilution to a higher dilution. Using the same pipette, transfer 1 milliliter of the 10 to the negative 1 to the remaining plate. Discard the pipette in disinfectant or appropriate container after use. The melted tryptic soy nutrient agar should be kept in a warm water bath between 45 and 50 degrees Celsius. The flask should not be too hot to hold in your hand. If the agar is too hot, it will kill the bacteria, and the agar will solidify in the flask if it is too cool. Wipe the outside of the vessel with a paper towel to remove any water. Gently swirl the nutrient agar, taking care not to make bubbles. Hold the flask plug with the same hand that lifts the plate lid. Using your pinky and ring finger, remove the flask plug, then use your thumb and forefinger to remove the lid from the plate. Pour 12 to 15 milliliters of the melted nutrient agar into one of the dishes so that it is about 1/3 full. Immediately replace the lid. Repeat this procedure to pour all 4 plates. After the plates have been poured, replug the flask and put the remaining agar aside. Gently swirl the poured plates to distribute the inoculum. Don't let the agar splash the lid or over the bottom. The plates should be swirled in a figure-8 pattern for approximately 60 seconds to thoroughly mix the agar with the diluted sample. Then stack all the plates to let them cool. Stacking plates reduces the condensation formed on the underside of each lid. Let the agar solidify. The solidified agar will be more opaque than the liquid. After the agar has solidified, incubate the plates, inverted at 35 to 37 degrees Celsius for at least 24 hours. Plate counts assume that each live bacterium grows to produce a single colony. In reality, some cells will stay clumped, so the results are reported as colony-forming units. The reason for plating serial dilutions is to reduce the number of colonies on a plate to a readable number. Remember, these all came from the same sample and would yield approximately the same number of colonies if you could count all the colony-forming units per milliliter, or per gram in the case of solid samples. Select a plate with between 25 and 250 colonies and count the colonies. Not all colonies will be on the surface because some bacteria were trapped in the agar as it solidified. These latter colonies will be noticeably smaller than the ones on the surface. However, all of the colonies on the selected plate must be counted. To obtain the count per gram or per milliliter of the original sample, the average number of colonies per plate is multiplied by the reciprocal of the dilution factor for those plates. The following formulas can be used in making calculations. Dilution factor equals initial dilution multiplied by subsequent dilutions multiplied by the amount plated, which is equal to 1 divided by the dilution factor multiplied by the number of colonies counted equals the colony-forming units per gram. After recording all of your data, properly discard your plates to be autoclaved.